Hello. Good morning, everyone. Thank you, much appreciated. So it's 11 o'clock, so we'll get started because you all want to get out of here by lunch, presumably. My name is Kael. I'm going to be taking you for neurophysiology and myophysiology, neuro being nerves and nervous system, myo being muscles. Last year, I was at this revision lecture, sort of just trying to take it all in. I remember it being really confusing to learn about, and sort of having somewhere that put it all together really made sense to me. So hopefully I can do that for you today. So these are the kind of things I'm going to be covering. So divisions of the nervous system, central nervous system, and peripheral nervous system with a focus on the autonomic nervous system. Sensory physiology, so receptors, ascending pathways, and nociception or pain. And then muscle physiology, so the types, what they do, and how they contract. So starting with the nervous system, as you've probably heard before, the nervous system is split into the central nervous system and peripheral nervous system. And then the peripheral nervous system can be split into motor and sensory. Sensory can be split into somatic and autonomic. And then autonomic can be split into sympathetic and parasympathetic. Oh, sorry, no, motor can be split into somatic and autonomic. This is a fantastic start. Okay, so here's an overview. So the central nervous system is the brain and the spinal cord, and also the eye, but you don't need to know that yet. So the brain receives sensory signals through the spinal cord and peripheral nervous system, processes those signals, and then initiates motor or efferent signals to the target organs. So most of the reception, processing, and initiation occurs in the brain. The only one of these that doesn't occur in the brain are the spinal reflexes. So those are within the spinal cord, and I'll talk about those later. So the brain is divided into two hemispheres, one on either side, and then each hemisphere has 4 lobes. And those are just named based on the bone they're under and where in the brain they are. So you have frontal lobe at the front, the parietal lobe on top, occipital at the base, and then the temporal lobe on either side. So it has regions within those lobes that are associated with very specific functions. And 2 of those are important for movement, and that's the primary motor cortex, which initiates voluntary movement, and the primary somatosensory cortex, which receives stimuli. So here are some of those areas. And those numbers are just Brodmann's areas. You don't need to know those yet. But these are the areas that are important in movement and sensation. So again, the primary motor cortex is involved in initiating voluntary movement. The premotor cortex plans out those movements. In the longitudinal fissure, you have the supplementary motor area, and that just coordinates bimanual movements. So if you're raising both your arms, that's what's going to be firing. The primary somatosensory cortex receives sensory information, and the parietal association cortex is just responsible for planning 3D movements. So if you're reaching out to pick up a cup and it's in a specific place in the room, that is the area that will be firing. So just a little bit more detail on the motor cortex and somatosensory cortex. So the primary motor cortex is found on the precentral gyrus, and the somatosensory cortex is found on the postcentral gyrus. And what that means is that the two bumps on either side of a fissure called the central gyrus. And it's called the central gyrus because it runs down pretty much the center of your brain. So the organization of those is contralateral. So the left side receives and sends signals to the right side and vice versa. Now, the amount of space on each of those areas is proportional to the dexterity and sensitivity of the area that it supplies. So that's called the homunculus map. And you'll see in an image of the homunculus, the hands are really, really large because they receive a lot of sensory information from the hands, and they also send a lot of fine motor output to the hands. So there's more space for them. Okay, so the cerebral cortex forms communication loops with the basal ganglia and the cerebellum. And you don't need to know these in a large amount of detail. You just need to know that it communicates with other parts of the brain. So in the basal ganglia, neurons will fire before a movement begins. So they are involved in planning movement, in skilled movements that you often do subconsciously, such as doing up a button, timing actions and scaling movements. So how large does this movement need to be? So that receives information directly from the cerebral cortex and sends the information back via the thalamus. And the basal ganglia is what's affected in conditions such as Parkinson's and Huntington's disease. So that really affects how you plan and carry out movements. The next functional loop is formed with the cerebellum. And the cerebellum contains a lot of our neurons, but it doesn't actually start any motions for itself. Instead, it modifies the signals that are going out. So it's involved in balance and posture, coordination, learning movements such as riding bikes, and cognitive function. So it receives information from the cerebral cortex via the pons and then sends it back via the thalamus. And the cerebellum is what's affected in cerebellar lesions, and it's why you get things such as dysdiadochokinesis, because it's really affecting your ability to coordinate those movements. So descending motor pathways from the cerebral cortex and the brain stem are divided into lateral and medial. So the lateral motor systems do your arms and legs, and the medial do your central muscles involved in balance and posture. And then the motor neurons that are sent out from the brain are divided into upper and lower. So upper motor neurons have their cell body in the cortex and brainstem and activate lower motor neurons. And lower motor neurons have their nerve bodies in the spine, and they directly innervate the skeletal muscle. And each muscle fiber is controlled by a branch from only one lower motor neuron, but one lower motor neuron can supply many different muscle fibers, and that's called a motor unit. Okay, so just to summarize motor signal transmission, the efferent signal is initiated in the primary motor cortex, which communicates with the basal ganglia and the medulla through loops. It descends down the spinal cord as an axon of an upper motor neuron. It crosses sides at the medulla and then travels along a descending pathway. And one of those descending pathways is the corticospinal tract. So the corticospinal tract, around 30 to 40% of those neurons originate in the motor cortex and 40% originate in the somatosensory cortex. That upper motor neuron then synapses with a lower motor neuron, which has its cell body in the ventral horn of the spinal cord, and then it exits as part of the spinal nerve to innervate its motor unit. So the organization of the spinal cord, I'm sure you've heard same Dave before, and that's just a way to remember what's coming in and what's going out. So sensory motor neurons arrive through the dorsal root and are afferent, so sensory afferent, dorsal afferent, and then motor neurons exit the spinal cord through the ventral root and are efferent, so motor efferent, ventral efferent. The spinal cord, as I said before, is capable of local reflexes called spinal reflexes, and those carry those out without needing to communicate with the brain. So those can be either monosynaptic with only one synapse involved, or polysynaptic, and the polysynaptic ones have an interneuron that can affect those movements. So There's a table with the spinal reflexes there. So the stretch reflex receives information from muscle spindles on the muscle length and the rate of change, and that's monosynaptic. The Gogi tendon reflex receives information from the Gogi tendon organ about tendon tension, and that's polysynaptic, so there's an interneuron. And then withdrawal receives information from pain receptors about mechanical and chemical stimuli, and that also has an interneuron. So here's a bit more detail on the stretch reflex. So the stretch reflex is the basis of testing reflexes in your peripheral nervous system exam. So if you suddenly stretch a muscle, for example, if you hit it with a tendon hammer, the muscle spindles will detect that stretch and then conduct an action potential to the spinal cord. In the spinal cord, they synapse directly with the alpha motor neurons, and then they conduct an action potential to the muscle and cause it to contract and resist being stretched. And that stops your muscle from being sort of stretched unawares to the point of damage. And that's why when you tap someone's knee with a tendon hammer, instead of nothing happening, it instead contracts and pulls their leg up. The Gogi tendon reflex stops avulsion and tearing of the muscle, and it does that by sort of monitoring how tense the tendon is and the rate that it's changing at. So if the pressure is too high or increasing too quickly, the Gogi tendon organs will detect that, send a signal to the spinal cord, and then synapse with an interneuron that sends information to the brain to let you know, hey, this is changing too fast, and also initiates an inhibitory signal to the fibers that that Gogi tendon organ is associated with. And the fibers around it will then equalize pressure, so you don't get tears in that bundle of muscle fibers. The withdrawal reflex is involved in like contact with painful stimulus. So if you touch something that's hot, you'll pull your hand away without even realizing that you've actually touched something hot yet. And that's because it's a spinal reflex. So the nociceptor will detect a stimulus, send an action potential to the dorsal root, where it'll synapse with an interneuron that again sends information to the brain, and then initiates excitatory signals to the flexor muscle. So that's the muscle that'll pull your hand away, and inhibitory signals to the extensor muscle to actually allow that flexor muscle to flex. Do we have any questions so far? Okay, so moving on to the peripheral nervous system. The peripheral nervous system is divided into somatic and autonomic. So somatic muscles contract. It's very voluntary, it's very specialized, it has one job and it's very good at doing it. So it only has one neurotransmitter, which is acetylcholine. It has one receptor, which is nicotinic, and it has one type of junction, which is neuromuscular. So it's pretty easy to remember. The autonomic nervous system is responsible for maintaining homeostasis. So all of the muscles and organs innervated by the autonomic nervous system have their own intrinsic function. If you cut out your autonomic nervous system, your heart would keep beating, your lungs would keep breathing, and you'd continue to digest food, but you wouldn't be able to change the rate that you're doing that at. And so that's why the autonomic nervous system is important for modulation of that activity. So the autonomic nervous system is split into sympathetic and parasympathetic. So sympathetic is fight or flight, and parasympathetic is rest and digest. So as opposed to the somatic nervous system, which has just one synapse, both divisions have two neurons in the peripheral circuit that communicate in a ganglion. So the preganglionic will have its cell body in the spinal cord and it's myelinated, so it sends a signal really quickly. And postganglionic has its cell body in the ganglion and it's unmyelinated, so it's much slower. And then the postganglionic neurons communicate with the cells of the tissue it's affecting. So your heart, your lungs, your intestines. So that table pretty much has an overview of everything, but a good way to remember it is to just remember parasympathetic and know that sympathetic does the opposite of that. So there are a few different ways for that. There's dumbbells, there's sludgeum. So this is a variation of that, be my sludge. So bradycardia and bronchoconstriction, so a slow heart rate and constricted bronchioles. Myasthenia, which is constrict sweating, lacrimation, which is the flow of tears and lethargy, urination, diarrhea, GIT, and emesis. So the parasympathetic and sympathetic nervous systems vary in where their ganglions are and also what type of neurotransmitter they use. So in the parasympathetic nervous system, the preganglionic axons are longer and the ganglia are usually on or near the target organ. And they have two synapses, both of them are cholinergic, both use acetylcholine. At the ganglion, the receptor is nicotinic, and at the target organ, the receptor is muscarinic. In the sympathetic nervous system, the ganglia is close to the spinal cord or about halfway. It has two different types of synapses. So at the ganglion, again, it's also nicotinic, but at the target organ, it's adrenergic. So it uses noradrenaline and it has either alpha or beta receptors. And here's just a diagram of that. You can tell this is American because it uses epinephrine. And that's essentially just showing where the synapses are and also what neurotransmitters are used in them. And here's the sort of flowchart for that. And that just goes over acetylcholine receptors, which are nicotinic and muscarinic, and then adrenergic receptors, which are alpha and beta. So there are a few different subtypes of these receptors, and you don't need to know these in all this detail. Just sort of know the first column. So M1 is neural, and you get salivation. M2 is cardiac, it's in the heart, and M3 is in glandular and smooth muscle and in the GIT. M4 and M5 are in this central nervous system. We're not entirely sure what they do. So just know that we're not sure about those. Adrenergic receptors are found in vascular smooth muscle, so in your blood vessels, and alpha-2 receptors are also found in presynaptic nerve terminals, and those inhibit neurotransmitter release. So if you activate those receptors, they'll then inhibit neurotransmitter release from that neuron. And then there are three types of beta receptors. Beta 1 in the heart, and it excites it. Beta 2 in the lungs, it relaxes them. A good way to remember that is you have one heart and two lungs. And then beta 3 is in the GIT. Okay, so in the autonomic nervous system, you have junctions. So the nerves will branch off in the infected tissue and have terminals spread out along their branches, known as varicosities. But there's a huge gap between those varicosities and the effector cell. And that means that you can release some neurotransmitter from that varicosity and it can affect a large surface area as opposed to just affecting one or two cells. So in cholinergic junctions, acetylcholine will cross the junction and any excess is removed by either acetylcholinesterase or reuptake transporters. So if you inhibit either of those, you get more acetylcholine. In the noradrenergic junction, again, it's a junction. Excess noradrenaline is removed by a reuptake transporter, which takes in excess noradrenaline, and then MAO and COMT will break down that noradrenaline within the presynaptic cell. Again, if you inhibit those, you get more noradrenaline available. So the sympathetic and parasympathetic nervous systems aren't competitive. You don't get competing between them saying, listen, I think my message is more important. If one's active, the other is usually inhibited. The exception to this is erections and ejaculations. So in these, they both function together. A way to remember that, point and shoot. P for point, P for parasympathetic. It acts on the penile artery using nitrous oxide, and that gives you an erection. And then the sympathetic nervous system, S for sympathetic, S for shoot, contracts the vas deferens for ejaculation. And Parkinson was weirdly excited about that. It was odd. Okay, so a few questions. At which level does the corticospinal tract decusate or cross over? Anyone have any ideas? Okay, so how do we want to do this? Do we want to have a sort of hands-up approach, or should I just show the answer? I'm hearing some C's. So The corticospinal tract crosses over the medulla, and that's just something you're going to have to memorize. I'm sorry. Okay, so next question. Which of the following is true? A, 40% of the corticospinal neurons originate in the somatosensory cortex. B, upper motor neurons connect directly with motor units. C, the spinothalamic tract decussates the medulla. Or D, A delta fibers carry slow pain. Give you a minute or so to think about that. Okay, does everyone have an answer? So the answer is A. I know that seems like a very random question, but it is something that could come up in your exam. And I'm very sorry about that. I know this is a bit of a random question. It could come up in your exam. Came up in mine. Okay. So which neurotransmitter is used between presynaptic and postsynaptic neurons in the parasympathetic nervous system? Acetylcholine, noradrenaline, nitrous oxide, or dopamine? I'm hearing some answers. So it's acetylcholine. And does anyone know what the receptor is? Is anyone brave enough? It's okay if you're not. It's a nicotinic receptor. All right, so same question. Instead, it's in the sympathetic nervous system. So is it acetylcholine, noradrenaline, nitrous oxide, or dopamine? Okay, so it's acetylcholine. So in the sympathetic nervous system, noradrenaline is used between the neuron and the target cell, whereas acetylcholine is used in the ganglia. Okay, you're cooking noodles because it's SWAT back and you don't have any food, and some boiling water gets on your hand. Which of the following would occur first? So this is talking about spinal reflexes. So A, the interneuron sends signal to the muscle to withdraw. B, a delta fiber sends a signal to the spinal cord. C, signal is transduced into an action potential. Or D, agonist muscle contracts. Okay, so I'm hearing some answers. So the first thing that needs to happen for you to feel anything or have any reflexes, a little bit of quiet. Pardon? So the first thing that needs to happen is that the signal needs to be transduced into an action potential. That can then be sent along the A delta fiber to the spinal cord. In the spinal cord, the A delta fiber will synapse with the interneuron. The interneuron will send a signal to the muscle to withdraw, and then the agonist muscle will contract. So the order this would happen is CBDA. No, CBAD, sorry. Okay. So, I don't know if you guys had this, but we had a very confusing lecture that sort of talked about the central nervous system, and there was a mention of the thalamus and some NTS, and I was like, okay, I don't know what that means. So the next 4 or so slides aren't really crucial information. They're more for your understanding, and there's something that you'll build on a bit in second year. So The autonomic nervous system is influenced by the central nervous system. And those influences are things like the thalamus, the hypothalamus, the cortex, and especially the medulla. So the medulla has control centers and a nucleus called the nucleus tractus solitarius. So the nucleus tractus solitarius, or NTS, is a series of sensory nuclei in the medulla. It forms the main input of sensory nerves of special relevance to the ANS. So these include things like baroreceptors, which detect a change in your blood pressure and then send that signal to the NTS. And then the NTS can signal your heart through the center involved in excitation of the heart, which is called the cardioexcitatory center. And that allows you to regulate your cardiovascular system. Similar function works for chemoreceptors that detect CO2 in the blood, and that's how it regulates respiratory systems. So I've got a couple of tables in the next 2 slides. This is sort of more for understanding what are different areas and how do they interact. The most important areas involved with the ANS are the NTS and the thalamus. So the thalamus is probably the most important input center of the brain. Most neurons will have their third order neuron body located in here. So most of your signals, most of your sensory signals will go up into the thalamus and then into, for example, the somatosensory cortex. The rest of those at this point are probably less important. And here's just where the NTS is located in the medulla, as well as some of its other centers. And you'll hear about some of these next year and you'll be like, oh, I'm pretty sure I've heard that before. But you don't really need to know them for your exam. And if you do, I'm so sorry. Okay, so just a brief word on emesis. The vomiting center, the area posterior in the medulla, is responsible for vomiting. So it's, which is above 1, 9, and 3. So that's responsible for vomiting. As I'm sure most of us know, vomiting is often preceded by profuse salivation, an elevated heart rate, and nausea. There are multiple triggers for vomiting from different centers in the brain and elsewhere in the body. So the cerebral cortex, where you get anticipatory emesis. So like if you think you're going to throw up, the cerebral cortex will be like, oh, well, I guess we're throwing up now. The vestibular center is involved in inner ear disorders and motion sickness. So if you get really dizzy and you throw up, it's because the vestibular center is like, listen, I don't think it's quite right. The chemoreceptor trigger zone is where drugs will work to make you throw up. And then peripheral receptors are involved in like ingested toxins, chemotherapy, and radiation therapy. So you don't need to know that in a huge amount of detail, just sort of understand that there's a vomiting center and there are several different centers that communicate with that. Okay, onto sensory physiology. So sensation is the detection of stimuli. Perception is the interpretation of sensory information. So there's a sensation of light on your cones and rods versus a perception of the image. Not all of our sensations are perceived. So baroreceptors, which modulate that monitor blood pressure, you don't perceive that as a sensation. You can't say, oh, you know, I think my blood pressure feels a little bit high today. Your baroreceptors do that entirely subconsciously. So there are 2 types of stimuli. The distal stimuli is the thing in the world, and the proximal stimuli is the physical phenomenon you get from that stimuli. So a light bulb is a thing in the world, but the proximal stimuli from that is light reflecting off your eyes. So sensory neurons detect the proximal stimuli and convert it into electrochemical signals or action potentials. So this is sort of, again, going over where the ganglions are found. So sensory neurons will have their ganglion in the dorsal root ganglion. And that's where you get your sensation. And then that'll send that action potential to the second order neuron, and which will send it to the third order neuron. And that's where you get your perception. So the type of receptor you have is determined by its ending. So chemoreceptors do chemicals, thermoreceptors do heat, nociceptors do pain, mechanoreceptors do vibration, touch, pressure, stretch, tension, photoreceptors do light, and proprioceptors figure out where your body is in relation to itself. So as I was saying before, receptors will detect a stimuli and convert it to action potentials, and that's called transduction. So transduction can encode different characteristics of a stimulus. So modality, what type is it? Locality, where is it? Intensity can be encoded by how frequent the fibers are firing or the number of fibers that are firing. And then the duration is encoded by phasic and tonic receptors. So nerve fibers can change their frequency of firing over time. Phasic adapt quickly. They generate a burst of action potential when they're first stimulated, then adapt, and then will release another burst when that stimulus is removed. So that indicates levels of change. So if you're going to touch your hand with a pen, for example, when you first put it down, they'll fire, and then when you take it off, they'll fire again. And that'll let you know it's been put on and then it's been removed. So tonic are slow adapting. They adapt slowly over time, and they tell you how long something will last for. So that period between putting it on and taking it off, those tonic receptors will always be firing. So that includes pain receptors and muscle spindles. So somatic sensation, this is touch. So touch is received from various different receptors: mechanoreceptors, thermoreceptors, pain, and limb position. So nociceptors and proprioceptors. Spatial resolution is how many sensory neurons there are in a particular area, and that varies in different regions of the bodies. So, for example, in your hands, you have a very high spatial resolution. There are lots of receptors in a very small surface area, and that means tests such as two-point discrimination will show that they can get quite close together before you begin to feel them as one. So the homunculus map and the somatosensory cortex relates to sensitivity. So the more sensitive it is, the more space it has dedicated to it in the cortex. There are different types of mechanoreceptors, and those are found on the two different types of skin. It varies between the two. So hairy or thin skin will have Purkinjean corpuscles and Ruffini corpuscles, whereas non-hairy or glabrous skin has all four types. So the depth varies based on their function. The closer they are to the surface, the better they are at detecting fine touch, and the deeper they are, the more responsible they are for pressure and grip strength. And their sensation is carried by A beta fibers. So here are the four types of mechanoreceptors. So there are two types of fast adapting and two types of slow adapting. And each of those types will have one with a small receptive field and high density, and one with a large receptive field and low density. So for the fast adapting, myasthenic corpuscles are involved in slip and grip. They have a small receptive field and a high density. And then Purkinjean corpuscles are involved in pressure, vibration, and touch. They have a large receptive field and a low density. For the slow adapting, you have Merkel's receptors. So those ones don't have a capsule, and that's the only way they really vary from the others. They have a small receptive field and high density. They perceive pattern and texture and shape. And then Ruffini's corpuscles are involved in stretch. So they have large receptive fields and low density. And that's just another diagram of those. And you can see how the fast adapting ones will fire at the beginning and end, whereas slow adapting will continue firing throughout. There are three types of receptors related to proprioception. So muscle spindles are stretch receptors that are parallel to muscle fibers. So when your muscle fibers stretch, those will also stretch. And then once they relax again, gamma motor neurons will tighten them to remove the slack, ready to stretch again. Gogi tendon organs that I covered earlier detect tension in muscle fibers, and then joint receptors detect movement in joints. Okay, ascending pathways. There are two main ascending pathways that you need to know about, and that's the DCML and the spinothalamic tract. So the dorsal column medial lemniscus pathway transmits proprioception, vibration, and fine touch. There are three orders of neurons. The first is in the dorsal root ganglion, the second is in the dorsal nuclei and travels to the thalamus, and then the third is in the thalamus and travels to the primary somatosensory cortex. So this decussates at the medulla, so at the brainstem. The spinothalamic tract transmits pain, temperature, and crude touch. So the first is also in the dorsal root ganglion, the second is in the dorsal horn, and the third is in the thalamus. it decussates immediately. So as soon as it enters the spinal cord, or about 1 or 2 levels above, it'll cross over to the other side. Okay, so nociception. This is pain. This lecture was pain. Nociception is perceiving pain, and it indicates injury. So physiological pain is important. It tells you when you've been hurt. But chronic or persistent pain is detrimental. It can be inflammatory, where you get inflammation, or neuropathic, where you have a nerve lesion. Clinical pain is sort of defined as when you have discomfort and abnormal sensitivity. So that can be dull, burning, or stabbing spontaneous sensation. And these are hyperalgesia, where you get exaggerated responses to noxious stimuli. So like if someone pricks you with a pin, it'll be painful, but it'll be really, really painful. Whereas allodynia is where pain is produced by normally innocuous stimuli. So even if you touch it with a cotton ball, you'll still experience pain. There are 4 different phases to pain or nociception. With all other forms of perception, you have transduction. So this nociceptors are activated and converted to an action potential. Transmission with this modulation, where descending neural systems edit pain signaling, and perception, where you become conscious to it. So pain is transmitted by A delta fibers and C fibers. A delta fibers carry fast pain. So something is hot, something is burning. You've been pricked with a pin because your partner in your CSSE was too harsh. So those are myelinated and have a medium diameter. They're myelinated so the signal can travel really quickly. C fibers carry slow pain, they're unmyelinated, so the signal travels much slower. So there's just a bit more detail on those. For A delta fibers, it's quite localized. They'll tell you exactly where the pain is, whereas C fibers are much more diffuse. A delta fibers feel noxious mechanical or thermal stimuli. C fibers feel noxious chemical stimuli mainly, and also persisting mechanical or thermal stimuli. So stuff like burns that you have for a long period of time. There are different types of these fibers. A delta fibers, there are 2 types. Type 1 is mostly mechanical, type 2 is mostly thermal. C fibers, there are a lot of different types. Their diversity is based on their peptide and ion channel expression, and I don't care about that, and you probably shouldn't either, because it's not necessarily for the exam. Just as an aside, the gate theory of pain. So C fibers will excite second order neurons and send a strong signal. A beta fibers will excite an inhibitory interneuron and inhibit that second order neuron. So A beta fibers carry information from mechanoreceptors, and activating those inhibitory interneurons by those fibers will inhibit the transmission of pain. It's why if you get a sore and then you rub it, it'll feel a bit better. So essentially, your C fibers will be like, listen, I think my message is important. A beta fibers come in saying, no, the thing that is touching my bruise, rubbing the skin of that is much more important than whatever message you have to send. I don't care, I'm inhibiting you through this interneuron. Sure, fine, go ahead, I guess. Okay, so you have the descending, descending modulation, and those work similarly to the gate theory of pain. So the neurons from the brain inhibit a second order neuron, and it's the same second order neuron that your A beta fibers will inhibit. So less firing means you have less pain. So the cortex, periaqueductal gray, you don't need to know what that is. Locus coeruleus, don't need to know what that is either. And nucleus breve magnus, again, don't need to know what that is, contribute to the pathway. And then the descending inhibitory neuron releases enkephalin, which inhibits the second order neuron. And the way that opioids work is they mimic enkephalin. This will be covered a bit more in pharmacology. These are sort of where different drugs will target descending and ascending pathways. So central sensitization is a form of long-lasting synaptic plasticity in the dorsal horn. It essentially just means that the nerves in the dorsal horn change to make things more painful. So the way that they do this is they reduce the threshold for firing, which means you get a progressive increase in pain. They increase the field size, so you get sensitivity beyond the site of injury. The neural firing patterns change, so you get hypersensitivity, and they recruit other neurons to the nociceptive pathway, which means you get a progressive increase. So this is the kind of thing that will happen in cases of chronic pain or long-term injury. And it's why over time, instead of things hurting less, they tend to start hurting more. So some questions. Which of these is found in hairy or thin skin? Okay, this is a bit of an unfair question. It's Pacinian corpuscles. So all 4 types of receptors are found in thick skin, but Pacinian corpuscles and Meissner's corpuscles are the only ones that are found in thin skin. Okay, where does the tract carrying vibration and proprioception decusate? So just try and remember which tract I said carried proprioception and vibration. Okay, was B. So the DCML carries vibration and proprioception, and it crosses over at medulla. Okay, so which of these is correct? Everyone have an answer? Okay, so the answer is B. The first thing that happens in any transmission of a sensory signal is always transduction. And that's the only one with transduction at the start, so it must be that one. Okay, we have a diagram. What color is the first order nociceptive neuron? Do we all have an idea? Red? Yeah, it's red. So where is its cell body? Feel free to call out, I don't mind. Dorsal ganglion? Yes. And where does it first synapse? Spinal cord, yeah. Do you know where in the spinal cord? Which horn? Dorsal horn, yeah. So the cell body is in the dorsal ganglion, and it first synapses in the dorsal horn. So which neuron has its cell body in the dorsal horn? Yeah, I'm hearing some seconds. So the first order neuron synapses with the second order neuron in the dorsal. So what effect will activation of the blue receptor have? Will it excite the first order, inhibit the second order? there can be more than one correct answer. Okay, so more than one correct answer. So it'll excite the inhibitory interneuron, and that will then indirectly inhibit the second order nociceptive neuron. So what receptor do we think this would be? This is just a bonus question. Okay, well, it's a mechanoreceptor. It's the Pacinian corpuscle because it looks a bit like an onion. So if you see an onion-like receptor in any of your histology slides, it's the Pacinian corpuscle. So what effect will excitation of the green neuron have? Okay, I'm hearing some Ds. Oh, I don't think that's quite correct. So it's D, and that should be different. I'm pretty sure that's D, and it's through the site of action for enkephalins or opioids. That one's my bad. So that actually is D. Okay, bonus question. Just in this specific diagram, the green neuron isn't synapsing with the inhibitory interneuron, it's synapsing directly with the second order. That's my bad, that's a mistake. Yeah, so, yes. Yeah, so I've folded the wrong one in this, and that's my bad. But the way this one differs from this one, the blue neuron is an A beta fiber, and that means that it will act on the inhibitory interneuron to then inhibit the second order nociceptive neuron. So that's an indirect inhibition. I folded the wrong one on this one, sorry about that. But what the green neuron, as a descending pathway, it acts on the second order nociceptive neuron to inhibit the ascending pathway through enkephalins. Yeah, I'll fix that in your slides. Okay, bonus question. What's the funky gray neuron doing? Okay, I'm running a bit short on time. So it's the spinal reflex. So it has the cell body in the ventral horn, which means it's a motor neuron. And because it's coming from a nociceptive neuron, it's a noxious stimulus, which means it's the withdrawal reflex. Okay, where does the spinothalamic tract decussate? Okay, everyone seems to be really on top of that one. It is the spinal level. All right, so we have 13 minutes for myophysiology. Let's take and go. Okay, so there are 3 types of muscles, skeletal, cardiac, and smooth. So both skeletal and cardiac muscle are striated. Skeletal muscle is voluntary. It has a neuromuscular junction. Cardiac and smooth muscle are both involuntary. They have intrinsic conduction and are modulated by the CNS. So the neuromuscular junction is found in skeletal muscle. It's where the efferent neuron innervates the muscle. It's a nicotinic receptor, so it's activated by acetylcholine, which is broken down by acetylcholinesterase. So in myasthenia gravis, there's a disease of the neuromuscular junction where you get autoimmune destruction of the acetylcholine receptors. And that's what will give you those symptoms of like muscle weakness and fatigue, because the efferent neuron really struggles to innervate that muscle because not enough of the acetylcholine is getting in time to actually innervate that muscle before it's gobbled up by the acetylcholinesterase. So treatment for that is inhibiting acetylcholinesterase, just to increase the amount of acetylcholine in that junction. To be honest, most of the drugs for like conduction issues where the receptors in the postsynaptic neuron are affected, they're just aimed at increasing the amount of neurotransmitter in that synapse. So there's a rule for you. Okay, skeletal muscle is made up of muscle fibers, and those are cells. So the muscle itself is wrapped in epimysium. The fascicle within the muscle is wrapped in perimysium, and the muscle fiber is wrapped in endomysium. So muscle fibers, or muscle cells, or myocytes are large and multinucleated. They have more than one nucleus. Their cell membrane is called a sarcolemma, and it has transverse or T tubules. So they penetrate into the center and mean that the impulse action potential can be conducted from the cell surface deep into the cell without having to travel through the cytoplasm. So within those myocytes are myofibrils. So there's a cylindrical groups of thick and thin filaments, and those thick and thin filaments are called sarcomeres. So the sarcoplasmic reticulum surrounds and it stores and releases intracellular calcium ions. And calcium is super important in the actual contraction of muscle because it needs to actually bind to a part of that sarcomere before the muscle can contract. So the sarcoplasmic reticulum is on either side of the T tubules and encompasses the myofibrils. So here's the sarcomere. It is the basic contractile unit of skeletal muscle, and it's what gives the muscle its striations. So they've got a sort of anatomy to them. So from one Z line to another, you have Z lines, which are zigzagging lines where the thin filaments converge. They are called Z lines. They look like a Z. I bands are the areas without thick filament. A bands are the areas with thick filament. The H zone is the zone without any thin filament, and it means that the actual sarcomere can contract without necessarily overlapping. And then the M line is the center. So in a sarcomere, there are thick and thin filaments. So the thin filament has three major molecules. So these are actin, which have myosin binding sites, tropomyosin, which covers the myosin binding site, and troponin. So troponin will bind to calcium, and then the troponin will cause the tropomyosin to retract and expose the myosin binding site on the actin. The thick filament is composed of myosin. It has a head and a tail that looks kind of like a hockey stick. So the tail points towards the M line, which is the middle line. And the head points towards the I bands, where there isn't any thick filament. Each head has an actin binding site and an ATPase site, and ATPase breaks down ATP into ADP and phosphate. So the cross-bridge cycle is what allows for contraction of muscle. So in order for the cross-bridge cycle to occur, ATP needs to bind to the ATPase site on the myosin head, where it's split into ADP and pi. And that is what sort of gives the muscle the energy to actually contract. So in the cross-bridge cycle, release of calcium from the sarcoplasmic reticulum exposes the binding site on the thin filament, and then the activated myosin can bind to actin. This causes the release of phosphate. So the myosin head will then pull the thin filament towards the center of the sarcomere, and this is called the power stroke. So this actually contracts the muscle. And then another ATP will bind to myosin and allow the myosin head to detach. And then the ATP will be hydrolyzed to ADP and phosphate, and that will activate the myosin head again. So rigor mortis occurs when you have the contraction of those muscles, but there isn't enough ATP to actually bind to the myosin head and detach it from that actin molecule. So instead of it contracting and then relaxing again, it will stay tightly contracted, and it means that you won't be able to pull it apart because all of the filaments are just contracted very closely together. So there are different types of contractions. Isometric, where you develop force but the length of the muscle doesn't change. So that's if you're trying to hold something in place, you're developing force in that muscle, but the muscle itself isn't changing in any length. Isotonic is where you develop a force and the length of the muscle does change. So concentric, the muscle shortens as it produces force. For example, if you're lifting up a dumbbell, your muscles will shorten to pull it closer to your body. And then eccentric is where the muscle lengthens as it produces force. So a good example of that is when you're going downhill, because it's working as a brake against gravity, which means even though it's contracting to stop you from falling, the muscle itself is stretching. There are 3 ways to vary the strength of a contraction. 1 is by muscle length, 2 is by frequency of excitation, and 3 is by the number of motor units you're activating. Muscle length, this is where length-force relations come in. So the force generated by the cross-bridge cycles during contraction relate to the degree of overlap. So in A, you have the optimal length. You can see that all of the cross bridges are overlapped, and there's also a space in the middle where you can actually move the thin filaments into, so you'll be able to contract that quite nicely. B is less than optimal length. There's an overlap of the thin filaments. The Z lines are hitting the thick filaments, and the cross bridges are misaligned. And that means that there's not really much place for you to go when you actually try and contract that. There's not a lot of room left. And then in C and D, there's less overlap of thin filaments. So in C, not all of the cross-bridge cycles are overlapped, and in D, almost none of them are. So you won't be able to contract that muscle very well at all. So the second is frequency of excitation. So each muscle twitch is the response to a single action potential. So the cross-bridge cycle will happen, and then the muscle will relax again. Summated contractions, while you're still responding to that first action potential, while the muscle is still contracting, a second action potential is produced, and then another contraction is created that can build on the first. So you get a slightly greater contraction. And then tetanic contractions is where you get repeated action potentials, and that means you have summated contractions that build into those tetanic contractions. So at low frequency, you'll get unfused, where you have that sort of waveform, and then at high frequencies, you'll get one very large contraction. So the third is motor unit activation. So as I mentioned before, a motor unit is a motor neuron and the fibers that it innervates. So you can have small or large motor units. So small motor units, it will innervate very few muscle fibers, and large, it will innervate a lot. So small, you get very fine movements, and large, you get very coarse and powerful movements. So if you have an action potential in a neuron, it will cause all of its motor units to contract, regardless of where they are, what they are, who they're doing, units will contract. The more motor neurons you have firing, the more motor units can contract, and that means you get more contraction. Is someone trapped? Whoa, I'll finish soon. So the smaller motor units are recruited first. Okay, questions and then we're done, I promise. Okay, so what is F? F is this whole line. It's all of this. It's the sarcomere. Okay, what is G? G is this section here. It's the I band, yeah. So it's where there isn't any thick filament. What is J? J is this section. Yeah, so J is the A band. What is I? I is this section here. It's the H zone. And here's the end. You're gonna be okay. be hanging around after, so let me know.